Hello, everybody, and welcome to a discussion of how we build our bodies. Often referred to as protein synthesis or transcription and translation, we'll be explaining how we get from the information in DNA to the bodies that put us together, and this is true for all living things. So last semester, we spent some time talking about how we can categorize and talk about all living things. And one of the interesting definitions is given by Alan Turing, stating that anything that can use the resources of the world to make copies of itself is alive. This is a pretty broad definition, and certainly one used by a computer guy makes sense in Alan Turing. But you can imagine a creature like Wall-E trying to make copies of itself, some robot running around a junkyard trying to figure out how to make more Wall-Es. Or you can imagine a plant trying to make more plants, or a human trying to make more humans. All of these are different ways we can be alive. And in the case of Wall-E, in our discussion last semester, we talked about really three different things or components necessary for something to make copies of itself. Obviously, a living thing is going to need the parts to build its body. If you're a Wall-E and you're trying to make another Wall-E, you need the parts to do it. So having those parts available is certainly important. You also need a plan for how to organize those parts. Somewhere in you as a robot or you as a tree or whatever, you're going to need to know how to organize those parts to make another one of yourself. And finally, you're going to need the energy to do this. You're going to need the energy to go around, pick up the parts, assemble them in the right order, and do whatever else is necessary to survive. Now, when we think of it in these very broad terms in terms of life, it sets up our discussion going forward. We spent a whole lot of last semester discussing that energy component. Living things use energy from the sun and the energy stored in bonds to do what's necessary to get themselves copied. The processes involved, of course, are ones we're familiar with. Photosynthesis and cellular respiration are how living things get energy from the sun or from one another to actually go through and build their bodies. Incidentally, this is also generally how they get a lot of their parts. We remember from photosynthesis, plants get many of the atoms they need from the air and water and other nutrients they absorb from the soil or the water that they're in. And we heterotrophs, like this cow here and myself, we get our parts from eating other things, the same source as our energy. But they're two separate things. We need the energy, and that's in the bonds between the atoms. And then, of course, we need the atoms themselves to arrange and build a cow. So I know it's a bizarre thing to say, but plants are made from air and water, and cows are made from plants. And it's just rearranging the parts that are in those organisms. And that's something we talked about to a large degree last semester. So if you need review, I'd suggest checking out one of those videos from last semester linked from your worksheet. But as we move on to this semester, last week we discussed the plan itself, or DNA. We now know that our plan, or the instructions for how to build our bodies, lives in this giant and fascinating molecule called DNA. This DNA is found inside the nuclei of our cells in eukaryotes like us and other mammals and plants and fungi, or it's found free-floating in the cytoplasm of cells in the case of bacteria and other prokaryotes. But this is a really important thing, that we have the information, we have the plan for how to build ourselves. But what we haven't discussed, the big sort of gap in our knowledge, is how we actually organize our parts according to the plan. We still need to discuss how we take those parts, the atoms necessary, and the DNA plan, and put them in the right order. How does the DNA talk to the atoms to get them to all put themselves together in the right order? We have a plan, we have the parts. How do we make a Wall-E? That's the thing we're going to talk about today. So our essential question, if you will, is how do living things use the recipe in DNA to assemble parts in the right way to build themselves? It's a complicated question, but it's an essential one to understanding how living things work. Yes, we have DNA. Yes, we have parts. But how does the DNA tell the parts to go together in the right order? This process is called protein synthesis, or affectionately transcription and translation. Now, in order to do this, we're going to launch off onto a really bizarre analogy for a little bit. So please excuse the analogy. I think it'll help our understanding when we come back to actual DNA and protein synthesis. So here we go. So for this analogy, we're going to be discussing making flan. What is flan? Flan is a fascinating and wonderful Mexican custard dessert. You should certainly try it if you haven't already. If you're lucky enough to have a family with cool recipes for flan, you'll know how good it is. But we're going to go ahead and discuss making flan today. So how do you make flan? So let's say Karen here wants to make some flan, right? Karen's the suburban mother living somewhere in the United States, and she wants to make the best flan possible to show her friends how great she is. And she's heard that the absolute best flan in the world is in Mexico in some grandmother's kitchen, because of course it is. The absolute perfect recipe, not found on the internet, not found anywhere except in this grandma's kitchen. And so Karen, willing to do anything to show her guests how amazing she is, she hatches a plan to go get this recipe from this grandmother in Mexico. I know this is bizarre, stick with me. So our recipe for flan is held by Grandma Dona Angelina. Doña Angelina has this amazing recipe. Thankfully, recently she's been on the internet. You should look her up, sharing all these great recipes online. But back in the good old days, she didn't do this. She kept all of her recipes to herself, but Karen got wind of it, and she wants to get this recipe. So there's a recipe for flan in Mexico that Karen wants. How's she going to get it? Well, first of all, we need to talk in some detail about this recipe, because Doña Angelina is artistic, and artistic people tend to be really eccentric. So she has this ridiculous middle school binder of recipes that she keeps her stuff in. She's not super organized like a lot of people. She's got this absolutely chaotic way of keeping all of her stuff together. Her recipe book has no organization whatsoever. It's not like desserts are in one section and then entrees in another and drinks in another. It's a jumbled mess, just like your best friend in middle school. Not only that, it doesn't just have recipes in it, it also has random other stuff, like photos of her granddaughter's christening and her niece's quinceañera. There's all of this random stuff in it. But every once in a while, you find this golden recipe, like her recipe for flan. Not only that, all of this is written in Spanish, which of course Karen doesn't read. Okay, so one more time. There's a recipe in Mexico in Grandma Doña Angelina's kitchen. It's in this bizarre middle school binder that's cluttered with other stuff. But somewhere in that binder is this jewel of a recipe to make the absolute perfect flan. So how's her recipe written? Again, Grandma Angelina is bizarre, and she's really eccentric, so she has weird ways of doing things. And she organized her recipe in tablespoons. Rather than saying get 2 cups of flour and 3 cups of water or whatever it is, she just works excessively in tablespoons. She's pretty artistic and eccentric. So all of her recipes read kind of weird. They're just a list of the ingredients each time she wants a tablespoon. So if she wants 3 tablespoons, she'd say crema, 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 because she wants 3 tablespoons of cream. If she wants vanilla, she'll say vanille, and just 1 tablespoon of vanilla, and whatever it is. And she just lists them this way. It's a bizarre way to have a recipe, but it works for her. And her recipe book is really disorganized, so it's even sometimes hard to tell when a story or some random thing stops and a recipe begins. But thankfully, the way that she organizes it, she actually has the word start, or comenzar in Spanish, when a recipe begins, and end, or terminar, when it ends. So all you have to do if you want to start a recipe is find the word comenzar, put every ingredient it has listed after that until you get to terminar, and then you have the right stuff. Of course, to make it even more complicated, sometimes in the middle of these recipes, she'll go off on a tangent and tell some other story. Something about a telenovela she just watched or whatever it is. But that said, the recipe's there. It's worth it. It's the best flan ever. This is how you're going to get it. So what's this recipe going to look like? Well, it's going to look something like this, right? Here it is. There's Grandma Doña Angelina's perfect recipe for flan. But looking at this, it's really hard to tell what's going on here. There are no spaces. There's no punctuation. It's a bit of a disaster. But somewhere in there is the magic recipe for how to actually make perfect flan. So let's dissect it a little bit. So first of all, remember, most of the stuff or a lot of the stuff inside her recipe book is not the recipe itself. There's lots of useless information, like all of the stuff here and all at the beginning and the end is all about some telenovela. It's not about making flan at all. But it's in there, and it's littering her binder, right? We only know that the recipe begins when we see the word comenzar, or start. So we know the recipe follows after that. So after the word comenzar, the recipe begins, and it's ingredient by ingredient listed however many times you need them. So if you take a look, and if you know your Spanish at all, you'll notice that the word azúcar repeats itself 4 times. That's 4 tablespoons of sugar. After that, huevo, huevo, 2 tablespoons of egg. Leche, leche, 2 tablespoons of milk, and so on. And it keeps on going. This is how the recipe is written out. And we know this recipe keeps going until we see the word terminar. But if you look, there's a spot in the middle of the recipe where we get this bizarre discussion about something else that has nothing to do with our recipe. It's again just about a quinceañera and her granddaughter's dress. So this is great, but it's not a recipe. It's in there, and then the recipe finishes off with more listed ingredients followed by the word terminar. I know, it's bizarre. It's a weird way to list a recipe. No one would do it this way on purpose except for an artist like Grandma Doña Angelina. But there it is. There's our recipe, right? It's all of those things listed with the word from comenzar to terminar minus that weird little story. So how do we get this recipe to Karen, right? So Karen, of course, lives in the U.S., and she doesn't speak or read any Spanish. We need somebody or someone to help us get that recipe from one spot to another and make it and put it in a format that Karen can understand. So not easy here, but it's certainly possible. But now you understand the plan. So what do we need? We need Pepe. Pepe is an amazing help in this process of getting the recipe from Grandma Doña Angelina to Karen. Pepe has some amazing skills that make him perfectly suited to this. To begin with, he speaks and writes both Spanish and English. Check, that's going to help because he can talk with Grandma Angelina and read her recipe, and he can also talk to Karen. He has a passport, so he can freely move across that border. Grandma Doña Angelina is just going to stay in Mexico. She doesn't want to go into the U.S., and Karen's not going into Mexico, so Pepe is our hero here. He can move across the borders. And importantly, he's related to both Grandma Doña Angelina and Karen, so he has no problem dealing with either of them. He's our hero. He's the one who's going to get things done. So Pepe is going to help us out. So what's he going to do? Well, okay. So Pepe, of course, has to go to Grandma Doña's house in order to get the recipe. He goes into her kitchen. He takes out a notebook, and what he does is he looks for that flan recipe. He looks for comenzar and writes whatever he finds until he sees terminar. He's going to write the entire recipe down and then take that eventually to Karen. So what does he do? So here's the recipe again, if you remember it. Pepe does what we call transcribing that recipe. He transcribe is like translate, but it's actually writing because he's going to write it down. So he transcribes the entire recipe from comenzar to terminar into English because he knows that Karen's going to read English. So as he writes it down, he rewrites the entire recipe, but writes it in English. So here you see the same recipe written, but this time going from Spanish to English. Now you'll remember that in the middle, there's this discussion about her granddaughter's dress. So what he also has to do is cut that out because he knows he has to write it down to be polite to Grandma Doña, but he doesn't need to bring it to Karen. But this last little piece right here, this is what he needs to take to Karen. It's the perfect recipe for flan from start to stop, written out in English. All right, so how does he do it? Well, once he has his recipe, again, he needs to go to Karen. So with that recipe, he crosses the border into Karen's kitchen and gets ready to go from there. Now, Karen's no idiot, right? She has all of her stuff ready to go. She's heard that Grandma Lupe's recipes are organized tablespoon by tablespoon. So she's got tablespoons ready of any possible ingredient. She just doesn't know how many to put together and what order to put them in. But she's got them ready, just like these ones you have here. So in order for this to work, what Pepe will do is he will read the recipe ingredients and Karen will organize those ingredients in the right order. So as you notice, he needs sugar 4 times. So he brings in sugar 4 times, followed by 2 eggs, then some milk, then sugar again, then cream, and so on. And we run out of space, but you get the idea here. This is how the recipe gets organized, tablespoon by tablespoon, and now we have the recipe to make flan. All Karen has to do is dump all those tablespoons together and mix them on up, and then we have our flan. And that's it. That's how flan is made. Okay. Thanks for bearing with us on this analogy. Let's write it all down so we understand it, and then hopefully it'll make sense why we're using it. So what are the steps? Well, again, number 1, Grandma Doña has a recipe for flan written in Spanish in her binder in Mexico. Recipes begin with the word start and end with the word end, and they're interrupted by these things, these weird little stories. Pepe goes into Mexico and transcribes, rewrites the recipe from Spanish to English, beginning with the word start and ending with the word end. The stories that interrupted the recipe are removed, and then Pepe leaves Mexico and travels to Karen's kitchen in the United States. In Karen's kitchen, the recipe is read word by word, and teaspoons are lined up with the words on one side and the ingredients on the other. Once the teaspoons are lined up, the ingredients are mixed together, making flan. There it is. That's our process. If you're working along on your worksheet, you'll notice you're expected to kind of list all of those in that table. So if you need to pause and write them down, you can do that. And now what we're going to do is talk about how this is actually very, very similar to how we go through the process of making a protein from DNA information. So let's change some terms around here. So this process, of course, isn't really making flan, it's protein synthesis. It's how we synthesize or make proteins with the information in our DNA. So this wealth of information that stays in Mexico, Grandma Doña, is really DNA. Like we said before, not a big surprise, DNA is where the information is stored. Recipes for proteins are actually what we call genes. So a gene is literally a recipe for a protein. You take a gene, you have all the information you need in order to make a protein. So again, not flan, but a protein. So DNA has a gene for protein. It's not written in Spanish, but it's written in the language of A's, G's, C's, and T's, which is the language we use to write in DNA. If you don't understand that, go back and review our earlier lesson. It's all in her binder, which is like one big chromosome or one big stretch of DNA, and located inside the nucleus of cells. Recipes, again, what is a recipe? Recipe is a gene, begins with the word start, and again, in DNA, this is literally true. They begin with what we call a start codon, which is 3 letters of DNA that literally means start in transcription. And they end with a stop codon, or the word stop. And then they are interrupted by these random stories. We call those random stories introns. Pepe is, of course, not a little kid with a backpack, but he's a really, really important molecule called messenger RNA, or mRNA for short. Those of you who've been paying attention to the news know how important mRNA is because it's what we're using for all of our vaccines for the coronavirus. So learning how mRNA builds proteins is exactly what we're talking about when we talk about the vaccine. mRNA is building proteins that will help protect us from the vaccine. Again, he goes into Mexico, but not Mexico, goes into the nucleus, and he transcribes the recipe from A's, G's, C's, and T's, which is the language of DNA, to A's, G's, C's, and U's, which is the language of RNA. You'll remember that A pairs with T in DNA. Well, A pairs with U in RNA. So there aren't any thymines in RNA. It's uracil that replaces it. And again, it begins with a start codon and ends with a stop codon. The stories that interrupt the recipe, again, are called introns, and those things are removed. So this step is no different. Then Pepe, or messenger RNA, leaves the nucleus and travels to Karen's kitchen. Karen's kitchen is where all of the ingredients are going to get assembled, and this is a really important organelle called a ribosome. So he leaves the nucleus, travels to a ribosome, and those ribosomes are located in the cytoplasm outside of Mexico. Finally, in that ribosome, the recipe, remember recipe is a gene, is read word by word. And in DNA, these words are what we call codons. A codon is a 3-letter word in DNA that literally means one of the ingredients to get. Just like azúcar, it would tell you to get sugar. CAG would tell you to get a certain amino acid, which we'll get to in just a second. So these words, or these codons, tell Karen which teaspoons to get. And the teaspoons are these really interesting molecules called transfer RNA, or tRNA. And tRNA lines up with these codons on one side and the ingredients on the other, the ingredients to make a protein. Now, the things that we put together to make a protein, you may remember from last semester, are called amino acids. So once these teaspoons come in, they line up, they drop off their ingredients or their amino acids, and then those amino acids aren't really mixed together, they're folded together, making a protein. That's our process. That's how we go through protein synthesis. Let me walk through that for you a little bit too, but again, it's a very similar one to what we just talked about with Grandma Doña and Pepe and Karen. The real protein synthesis, of course, happens inside a cell. First of all, this is a eukaryotic cell. It has a nucleus, and that makes it different from bacteria cells. What separates the cell from the outside world is called the cell membrane. And what we call the inside of the cell, the jelly-like water structure inside, is called cytoplasm. So we've got the thing that separates the cell, the membrane, and then the cytoplasm. Inside the cell, we have this darker circle called the nucleus. And the thing, of course, that separates the nucleus from the rest of the cell is the nuclear membrane. And inside the nucleus, we have these really important molecules called DNA, organized in chromosomes. And finally, the last organelle we care about for this process are these little dots out here called ribosomes. But this is where everything happens when we make a protein. We use the information in the DNA to build a protein in the ribosomes. So again, our nucleus is like Mexico. Our gene is that recipe somewhere on this chromosome. But again, it's not written in Spanish. It's, of course, written in the language of genes. So let's transition over from our analogy to kind of the real-world example. And here would be a recipe written in the language of DNA. Now remember, it still has all those components. The useless parts, the word start, the word stop, the story in between. All of those are in there. We just have to be able to recognize them. So let's start breaking it down. Well, remember, we're looking for the word start and the word stop in our gene, in our recipe here. And it turns out the word start in DNA is ATG. We call it a start codon. And there's several stop codons, but one is TAA. And all of these tell us when we start and when we stop our process of copying that gene. Now, everything before and after that is not useful. It's stories about novellas, quinceañeras, and so on. So we don't care about that. What we care about are all of the little combinations inside it. Remember, there's also that tiny little bit, the grayed out part here, that's an interruption inside the gene itself, and sometimes we get those as well. But what's important to us are all of these codons, these three-letter words that are going to tell us what ingredients to get when we actually build our protein. So there's our gene minus the useless part that's sitting in the middle there. So here's that same gene laid out for you in DNA. When RNA actually goes through, what it does is it untwists that ladder of DNA and it gets this long strand of DNA from start to stop, and it begins what we call transcribing. Again, think of it as Pepe writing down these new ingredients in his notebook. And literally what happens is Pepe comes in and transcribes that one letter at a time, bringing in the right pairs for each of the letters in the recipe. So here he goes. He pairs up every single letter of DNA with a letter from RNA. Every T gets an A, every A gets a U, every G a C, and every C a G. And he transcribes the entire gene, including that useless part, of messenger RNA. And this is what's going to leave the nucleus and be ready to go into the ribosome to actually make the protein. Once he has his strand of messenger RNA built, he's ready to go. He's just got to cut out that useless part, which he does here, and then restitch the RNA together. But now he has this completely useful, written in the language of RNA gene that he can leave the nucleus with. And that's what he does. Once he has this all ready to go, he leaves the nucleus and heads out into the cytoplasm. So once he leaves the nucleus, good old Mexico here, by crossing the nuclear membrane or the border between Mexico and the US, he comes into the cytoplasm or the United States. Now, of course, what he's most interested in is going into Karen's kitchen, which is a ribosome. All he has to do is leave that nucleus, come out into the cytoplasm, and then get pulled through the ribosome. As he gets pulled through the ribosome, every single one of those codons is going to be read, resulting in the actual ingredient. So how do we get the ingredients? We use transfer RNA. Now remember, transfer RNA are like these tablespoons, with a word listed on one side and the actual ingredient on the other. But of course they're not cream and milk and vanilla and sugar, they're amino acids. Transfer RNA molecules are set up just like this. On one side, they have what we call an anticodon. It's the opposite of the word that's listed on the messenger RNA. We'll explain that in a little more detail in a second. On the other side, they have the actual ingredient, the amino acid that's going to get chained together to make the protein. So anticodon on one side, amino acid on the other, and this is true for all of them. So there's lots and lots of tablespoons, or lots and lots of transfer RNA molecules, each with a different anticodon and a different amino acid. So how does it work? Well, here we are, ready to go. We've got our transfer RNA molecules with their anticodons, and we've got the stretch of messenger RNA with the codons on it. That stretch of messenger RNA gets pulled into the ribosome, and what happens is that UAC, if you know your genes of messenger RNA, UAC pairs with AUG, U with A with U, and C with G. And that's going to be the very first amino acid dropped off. And yes, it's methylalanine. It's that start codon that gets dropped off, and we get the very beginning of our protein. Then what happens is that little strand of messenger RNA keeps getting pulled through, and we get the next codon. In this case, it's GCU, which pairs up with CGA, and that codes for the amino acid proline. Proline comes in, gets dropped off, bonds with that methylalanine, and this process continues. Another strand gets pulled through, the next pair gets brought over, and it paired up with AAU, drops off another amino acid, and this keeps going. Another strand gets pulled through, CCU, GGA, drops off another amino acid, and there we go. And this continues as the entire molecule gets pulled through. As the molecule gets pulled through more and more, we get this entire long strand of amino acids. And once we have this long strand of amino acids, it's ready to get folded up to make a protein. And it's a bit of a complicated process, but the thing literally folds up, building the protein or finishing off our process and making the flan. So once again, protein synthesis, we have the information in DNA, it gets transcribed by RNA, that RNA leaves the nucleus, and then codon by codon, amino acids get dropped off, which make the protein as they fold together. This is protein synthesis. It's confusing, but it's super important in understanding how organisms build their bodies.